I appreciate the Bob Langer commercial. I agree, he is absolutely fabulous. This is our forward-looking statement, safe harbor clause. You guys have all seen those before. I want to talk a little bit about our ocular programs. The company has a diverse set of cells that we can create using human embryonic stem cells as the progenitor cells. But in particular today, we're going to primarily focus on the use of those cells and then the cells that we then culture from them, focused on the ocular program. Our leading program by far, of course, is the use of retinal pigment epithelial cells, which is the direct cause of numerous different kinds of blindness. The two indications that we're working on particularly right now are age-related macular degeneration, which, as you heard from Marty earlier today, is the leading cause of blindness in people over 55 years old. And our second indication is an orphan indication called Stargardt's macular dystrophy, which is a highly unfortunate juvenile form of the disease. We create these RPE cells out of the embryonic stem cells, and the RPE cells provide a whole bunch of different factors to the back of the macula. I'll talk about that in more detail later. But I want to talk about a couple of other things that we are doing in the back of the eye. On the hemangioblast side, so we take hemangioblasts. Hemangioblasts are the precursor cells for all of the endothelial and hematopoietic cells in the body. Now, one of the things that is great about hemangioblast cells is that they can create angiogenesis. So they can solve, we've seen, for example, in our animal models, we've seen them solving ischemic limbs and diabetic retinopathy. So we can use these cells in the eye to look at ischemic retinopathy, including diabetic retinopathy. We've also created retinal neural progenitor cells. These are the cells that can become the photoreceptors, rods, and cones. We've started using those in some animal studies, and we've seen some really remarkable things happen. Now, the exciting thing about the potential combination therapy here would be doing something with the RPE layer and then us going in and perhaps doing something where we can create some photoreceptor progenitor cell so that we could maybe, in combination therapy, treat patients whose disease is very, very far advanced. We've also done some exciting things on the corneal side. We've created corneal endothelium and epithelial tissue, as well as Descemet's membrane. There is a very large unmet market of need for corneal transplantation in this country, and particularly in other countries where transplantation after death and organ donation is not a very common practice. And so the way that corneal membrane is typically gathered is through cadaver. So this is an exciting new field for us as well. And we've also created mesenchymal stromal cells, and there are a whole bunch of different ways that we can use these, but our particular indication that we're looking at right now are both glaucoma and uveitis. I want to talk a little bit about retinal pigment epithelial cells. These are the cells that we directly culture and terminally differentiate. And these are critical to the function of the back of the eye. These RPE cells recycle photopigments. They provide phagocytosis to the back of the eye. They provide an entire protein and vitamin distribution network for the back of the eye. Essentially, what they feed is not only what's called the Brooks membrane, but quite importantly, in the early stages of the decline of blindness, they feed these photoreceptor cells, the rods and cones. And as you observe the retinal pigment cells begin to decay and ultimately die, what you see is a corresponding loss of the photoreceptor layer. I'm going to show you that in a little bit more detail through some amazing imagery that we've been able to do to the back of the eye. The RPE loss, like I said, leads to this photoreceptor loss, which leads to a whole bunch of different kinds of blindness, the leading cause of which is age-related macular degeneration. We've also filed, as I said, on Stargardt's, and the next IND we intend to file is for myopic macular dystrophy, which is a distension in the back of the macula, which causes fissures and a breakdown of the RPE layer. It's an enormous target market, and particularly enormous in the Asian market sector. The RPE cells are very interesting because there are only two kinds of cells in the body which are pigmented. Now, this gives us a very significant advantage. One of the things that the FDA was very focused on was making sure that we're not going to inject any non-terminally differentiated cells into the back of the eye, in particular, any undifferentiated embryonic stem cells. In the treatment of our 168 animals that we treated in the preclinical work, and of course in the 2 humans that we've treated so far, we've observed no ectopic tissue, no tumor formation, no significant adverse events. in the use of these cells, even when we spike these cells with a small amount, up to 1%, of undifferentiated human embryonic stem cells. However, when you inject human embryonic stem cells into the back of the retinal space, you are virtually certain to create tumorigenicity and significant ectopic tissue. Now, one of the great things about having this tissue be pigmented like it is it makes the assay for identification of these cells much more efficient than the standard PCR marking. In fact, we believe that our assay is about 20 to 30,000 times more efficient than the traditional PCR marking for identifying these cells. And that's one of the critical aspects that the agency was focused on, again, on this issue of not wanting to inject any non-terminally differentiated cells into the back of the retina. We targeted the eye in general because in a healthy eye where the back of the Brooks membrane is not compromised and there's been no vascularization of the back of the eye, you've got a generally immune-privileged center. So we're using an allogeneic source of cells here, which allows us to create significant batches of these cells, freeze them down, and ship them out. So it looks like a monoclonal antibody or some traditional pharma. We see this as a very scalable model and something that's very critical in terms of rolling out the commercialization of this product. The ease of administration will show you the cannula a little later, but all of the materials we use for the injection are off-the-shelf cannular devices. We have a GMP manufacturing facility in Marlborough, Mass. At our lab. We are capable of making about 500,000 doses a year of these RPE cells. Essentially, if you think about the amount of cells we're injecting in the suspension of fluid that they're in, it's about the head of a pen. So you're talking about a very small number of cells necessary. The back of a healthy eye on the Brooks membrane layer within the fovea or the central field of vision is only about 60,000 RPE cells. So we don't really need that many cells to be able to recreate what's in the back of an otherwise healthy eye. Obviously, one of the targets that we're working toward is centralized manufacturing, stability and ease of manufacturing. Now, obviously, when you're in the early days of building these cells, there's a lot of high touch involved. You want these cells to have the right amount of pigmentation and a variety of other factors, and we feel that now we've really kind of built a process for making that scalable and repeatable. This is an example of what we saw in the, I don't know if there's any pointer on this, but at the back of the mouse eye, there is this Brooks membrane here, and you can see there are these empty spaces where the mouse RPE layer has gone into complete decline. And so on the injection site, which is right around here, on the injection site, the cells then migrated. Not only did they migrate across and sit down in a place where there was largely no mouse RPE layer, but they actually skipped across a place where there was already some healthy RPE layer. So through some immune technology and this fluorescence that we use, we're able to identify where these cells laid down. Let me skip to the next slide here. What you can see is in the animal, in the treated eye and the control eye, the result is that you can maintain a very stable layer of photoreceptors. You can see in this treated eye, it's about 5 to 7 cell widths thick photoreceptors. The same animal in the same time period that's untreated, you can see a very unstable and unhealthy photoreceptor layer. Perfect, no problem. So you can see that we've had significant effect in the animals. This is the design of our trial. It's 4 cohorts of 3 patients each. We were treating both Stargardt's and AMD. The first patient in Stargardt's that we treated was 26 years old. She's now 27. She had best corrected visual acuity to 0 letters on a standard eye chart, and the 77-year-old had BCVA of 20 by 500. The surgical technique is relatively very simple. It's a 25-gauge pars plana vitrectomy, followed by the injection of the cells, an air fluid exchange, and then the bleb goes down. I'm actually going to show you the surgery here. On the upper right, you can see the cannula device that's used. It tapers down to about 39 gauge, and it's got a soft tip so we don't penetrate the edge of the choroid. Here you can see the surgery taking place. We remove the gel from the inner surface of the retina. It's a very short surgery, actually. And you'll see the bleb come up now in just a second. Okay, that's the injection of the RPE cells into the bleb. And then we're going to switch the angle a little bit here. And there's an air fluid exchange. This is done at the intersection of a relatively healthier part of the retina outside the fovea and the atrophic part. Here's the air fluid exchange. So the patients are, once the surgery is over, and you've just seen the entire length of the surgery effectively, once the surgery is over, the patients are wheeled on their bed. By the way, they're awake for the whole surgery. The patients are wheeled out, and they're kept observed for a couple hours. The bleb mostly was dissolved after about 2 hours, and we kept them overnight for observation, although in the ordinary course we don't obviously expect to do that. The patients are treated with a mild course of immune suppressant, although we ultimately think that the clinical use will not necessarily require immune suppression because of the immune privileged nature of the back of the eye. I talked a little bit about our other programs. I can sort of skip over them. But the one important thing about the company is that the traditional method for harvesting human embryonic stem cells involves the removal of the inner cell mass of the embryo, thereby destroying the embryo. We don't do that. We extract a single cell from a much earlier stage morula, a 4 to 8 cell morula. We do not change the fate of the morula. We do not destroy the morula. So there are some religious and ethical as well as some very significant scientific advantages to that as well. In terms of intellectual property, we've got very broad intellectual property coverage around not only culturing the cells from embryonic stem cells, but also for the clinical use of these kinds of RPE cells. And in terms of the company, we have a market cap of about $275 million, and we ended the year with about $13 or $14 million in cash on the balance sheet. Our burn rate is about $3.5 million a quarter, so we're funded for 2012.